Hi dear students, let's continue with the second part of the spoilage of sugar-based products. In the previous section, we had come across the spoilage which has been caused to chocolates. Now let's concentrate on sugars. Sugars, as we all know, is generally being considered to be as sucrose. And sucrose is mainly derived from two primary agricultural sources. One is sugarcane called Saccharum officinarum, as well as there is another one called the sugar beets, referred to as Beta vulgaris. Now, in subtropical as well as the tropical climates, sugarcane serves as a main source of sucrose, whereas in the moderate climate regions, sugar beet will serve as a source of sugar or sucrose. And these two sugars will represent about 99% of the world's sucrose production. And we can see that a small amount of sucrose is also being produced from sugar palms, as well as sweet sorghum, as well as from the maple trees, which have been found to be located in different regions. And as we have mentioned earlier itself, a sugar, the term sugar does not always implies that it is sucrose, though sucrose is the most widely used sugar that we have. We also do use various other sugars such as dextrose, lactose, fructose, etc. as a type of sugar. And more recently, we can see that various sugar-free confectionaries as well as chocolate products will contain polyols such as sorbitol, mannitol, xylitol and maltitol as a replacement of sucrose in the sugar-free components. So in all these, when you refer to sugary components, we refer to all these sugars which can be used in the food. Now, to deal with the entire type of sugars is not something beyond the scope, so I'm just concentrating on the spoilage of sugarcane based sucrose. As we all know, the processing of cane sugar mainly involves different steps. You harvest the sugar cane, you crush and extract the sugarcane juice, you clarify it, you evaporate it and you crystallize the sugar from it. You centrifuge and take the crystals, you take the raw sugar and you further refine it. And you know the raw sugar has a different color, and when you refine it, you get it as a white sugar that we consume right now. So, at different stages of this sugar processing, there are chances of spoilage of sugar by microbes. And the thing which we should note is that sucrose is a good source for the multiplication of microbes. And with the carbon source, the microbes can rapidly grow. So, at every step, we should try to minimize the amount of microbe exposure. Now, how can you say that you cannot do the process of each and every step of sugarcane processing in a sterile environment, because it happens in the nature. And at different stages of processing, the sugarcane is being prone to different levels of microbial spoilages. Now, if you go to consider the pre-harvest, you can have Flavobacterium, Lactobacillus, Xanthomonas, Enterobacter, Pseudomonas, Erwinia, Leuconostoc, Bacillus, Corynebacterium, yeast as well as molds. And after the harvesting, you can have Leuconostoc, and crushing and extraction will be involved again Leuconostocs. But when you have a clarification, it's not much of microbes which have been found. But when the evaporation, crystallization and centrifugation is being done, the predominance of thermophilic spore formers are mainly being found in the evaporation as well as the following refining steps. So the ultimate aim of a sugar producing food industry is to limit the spoilage which could be caused by these various microbes. Now, how do we control this? As I told you earlier, it's totally impractical to take the sugarcane and leave it into a completely sterile environment, because that is not possible. But you can see that you can somehow reduce these microbial spores or control these microbes to an effective level, so that they do not grow in a very large population and cause ultimate spoilage of the entire sugar. Populations of Leuconostoc nematodus, which may increase during the harvesting of sugarcane, may result from contact with contaminating harvesting tools as well as insects. And this organism, it can hydrolyze the sucrose to dextran, which has been found to be a slimy, gummy as well as a glucose polymer. So, an effective cleaning and sanitation program for the mill equipment will help us to control the presence of these microbes and also reduce the contamination from the raw juice. Another option that we can have is you can heat the juice from about 80 to 100 degrees centigrade, and that would kill almost all the vegetative bacteria as well as the yeast. But you have to make sure that, keep it in your mind, that only bacterial spores like Bacillus species as well as Clostridium species will remain in the raw juice. And the microbiological specifications for different types of sugars mainly include, there is a specification which has been already been provided. You can see that if you're having sugar, the total thermophilic spore count of 5 samples from a lot of sugar, none shall contain more than 150 spores per 10 gram, and the average for all the samples shall not exceed to 125 spores per 10 gram. So the chance of reducing the spoilage is to limit the spore count to so much level. You cannot say that it will be completely sterile, but you have to limit the count of the thermophilic spore forming bacteria to this level. If it is not beyond this, you need to do a sterilization technique or something which would prevent. Of course, you have to keep in your mind, you cannot give excess heating over here, because if you give excess heating, what happens? The sugar will melt and sometimes it would get even charred. You cannot give, it's before the crystallization step that you have to do this sterilization, because the raw sugar can only be, the raw juice, remember that, okay, the raw juice can only be heated here to 80 to 100 degrees centigrade. And the flat sour spores of all 5 samples which have been taken, none shall contain more than 75 spores per 10 gram. And coming to the thermophilic anaerobic spores, not more than 3 out of 5 samples shall contain these spores. And the sulfide spoilaging spores, not more than 2. And if you're going to deal with bottler's granulated sugar, mesophilic bacteria should not be more than 200 per 10 gram, yeast not more than 10 per 10 gram, malts not more than 10 per 10 gram. You just have to have a basic outline of how much is permitted. And beyond that, the industry is not allowed, such products are not being allowed to be marketed, because that would get spoiled. And if you're going to the bottled liquid sugar, you can find the mesophilic bacteria. If you're going to take last 20 samples, should average 100 organisms or less than 10 gram of the dry sugar equivalent. The yeast, the last 20 samples should average 10 organisms or less per 10 gram of the dry sugar equivalent, as well as the malts, the specifications are same as that of the yeasts. So I hope this is clear to you, the sugary components. The sugar, this is almost applicable with the beet sugar also, but I'm not going into the details of that in an expanded way. Now you just keep in mind, you have to reduce the microbial spoilage, and this is possible by treating the raw sugar or the raw juice, okay, at a temperature about 80 to 100 degrees centigrade, so that the level of vegetative bacteria as well as the yeast will be reduced, and but what remains could be the spore forming bacillus as well as the other Clostridium species. Thank you for now, students, for your attention. Hope so this is clear to you.